thanks for the opportunity to speak today. I've been in the Nanopore business since 1996, so I'm really honored to be presenting for our community. I'm a disclaimer to begin with, some business items. I am a shareholder and consultant to Oxford Nanopore Technologies. I'm going to be relatively positive about the Nanopore platform because I'm convinced it's going to contribute substantially to genomics in the now and in the future. But there are some issues that we can improve upon, and I'd open the question session to all of you to pursue some of these areas for improvement. Whenever I give talks of this sort, I have two major audiences. One are undergraduate students, and at the other end of the spectrum are people who I characterize as the smartest person in the room. Oftentimes they're physicists. So there'll be parts of my talk that for molecular biologists and nanoscience experts are redundant. But for a new audience, it's important to touch base on those. This is a self-portrait of Da Vinci, for example. All right, so my outline is I'll talk a little bit about speaking with Silicon Valley about DNA sequencing. Our campus is about 45 minutes from Apple headquarters, so we're fairly familiar with the comings and goings in Silicon Valley. I'll talk about the invention of nanopore sequencing and some early experiments that have people have forgotten about that were pivotal. And then I'll talk about the meat of my presentation on nanopore sequencing presently and in the future. Silicon Valley appreciates large numbers, as you might imagine, more features on a chip, billions of dollars of investments, and so forth and so on. And so I wanted early on to impress upon these essentially CS folks and physicists in Silicon Valley about DNA, its power. And there was a study done, I think it was by a group in the Netherlands or Denmark, I apologize for not remembering, and they estimated that we're on the order of 10 to the 37th bases of DNA on planet Earth. And even if they're off by 5 orders of magnitude, this is an enormous number. And to give you an illustration, if you were to stack the DNA bases 3.4 angstroms and to basically take all of this sequence and align it up, you'd go back and forth across the Milky Way 6 times. So there's a lot of DNA to be explored. And as long as there's sentient beings on this planet, they will be improving sequencing technology. So a little bit, I am old enough to know a time before Sanger sequencing, and you may all be shocked that such a thing exists, but it did. And so in 1976, for example, sequencing was essentially one base at a time using p32 labels. And so in 24 hours, you could generate a single nucleotide of sequence, which was dreadfully slow. Along came Sanger in 1977 with probably the most influential technology for the past 30 years in biology. And so credit to him. I think the first real foray into human genetics was the mitochondrial genome, which was sequenced using this technique in 1982. Mark went through this figure. So Sanger sequencing originally would have cost way too much to assemble a human genome, and this dropped down through robotics and CS and linear polyacrylamide, for example, to about $100 million, as he showed before. So he already went through this part of the slide. I don't need to belabor that, the advent of massively parallel sequencing, Illumina, Solexa. But what I wanted to emphasize, and Mark did this as well, is that now this is dropping down to, some people estimate, about $100 per genome for materials, at least. And I was a history major back in the day and minored in molecular biology, but my history degree taught me to think long-term. And so to date, we've reduced the cost by 4 orders of magnitude, and to get to $1 per genome is 2 orders of magnitude. So easily in the careers of undergraduates on this call, the cost I predict, and I think others would agree, is going to be approaching $1 per genome. Nanopore sequencing, the invention was in 1989. David Deamer pulled over to the side of a road in Oregon, a 2-lane highway, with this idea and wrote it down in his notebook. This is a diagram from that notebook. At the same time, George Church in Boston at Harvard came up with a parallel idea, and the two of them were joined by Dan Branton a year or two later for this fundamental technology. By the way, NHGRI has sponsored and supported this, NIH and then NHGRI, for more than 2 decades. So NHGRI deserves a lot of credit for its implementation. So many of us were skeptical about this technology when it was first conceived in 1989. I included, but there were reasons for optimism. And one is that it's a straightforward concept. It's basically a molecular culture counter. So on the left, you see a protein channel in a thin film, originally a lipid bilayer. You add salt. And then in the middle frame, you apply a potential and you get current carried by potassium and chloride ions. And polyanions like DNA or RNA are captured in that electric field, and it causes an impedance in the current that one hoped at the time would be indicative of the base identity going through the pore. Another reason for optimism is there's a very large electric field. So electric field is volts per centimeter. So if you apply 200 millivolts to a field, an aperture in a pore that's about 100 angstroms long, you do the math and it's hundreds of thousands of volts per centimeter. And if you take a 2 molar KCl salt solution and 200 millivolts applied potential, that gives you about 10 to the 20th particles per millimeter squared per second. That's a big number, and for you to understand that on a macroscopic scale, if you scale that flux up to something we can perceive, it's basically a lightning bolt. So there's a very strong field and a lot of flux through this nanopore. The meaning of that is that if you slow down progression of the DNA from microsecond time scale to millisecond time scale using an enzyme, that comes out to about 1 million to 10 million ions probing each nucleotide. And lastly, the other reason for optimism is that the electronics at the heart of this technology is time tested. This is what's called a voltage clamp amplifier. I'm not going to talk about it a lot because I haven't got time, but it was invented in 1947. And to give you sort of a reference point, Hodgkin and Huxley described basically the phenomenon where you can apply a potential to a neuron and watch it migrate along that neuron. That discovery, that action potential, was relied upon a voltage clamp amplifier. And there have been at least 2 other Nobel Prizes that were founded on this electronics. So an early pivotal set of experiments were capture and translocation, and that needed to be shown. And my colleague, Dave Deamer, formed an alliance with John Kasianowicz at the National Institutes of Standard and Technology in Maryland. And John had optimized a protein pore called alpha hemolysin to be stabilized in thin films. And so Dave and John put these together in a patch clamp device, applied a potential, and added polymers of RNA to the system. And what they saw were downward deflections of the ionic current associated with presence of these polymers, as depicted here. So the question then at the time was, are those collisions are those capture events due to a collision, as depicted on the left side, or are those due to translocation on the right side? And I, among others, were convinced that it was most likely to be collisions as opposed to translocation. And so Dan Branton came up with this decisive experiment, which is depicted here. So there is the cis compartment where you add DNA or RNA strands, and the trans compartment to which these strands may be translocated. The line in the middle is a depiction of a thin film, a lipid bilayer, and there are channels in that thin film through which the DNA or RNA could be translocated. So they devised a combination of single strands that they thought could go through the pore, depicted here, with sequences that could be specifically amplified. And as a negative control, they included duplex DNA, again, that could be amplified specifically, that did not go through the pore. So they applied a potential, counted translocations, and used qPCR to see if those translocations corresponded to the single strands in the trans compartment. And the result of that fundamental study was that, in fact, it was translocation. So this is a PCR gel of PCR of these fragments. So in the cis compartment, you saw both double-stranded DNA and single-stranded DNA. And in the trans compartment, the only strands that amplified were for the single strand. And what is more, the number of blockades that were counted per minute on a nanopore were essentially equivalent to the amount of molecules in the trans compartment measured by qPCR. So here, just to take it a step further, we then knew that individual strands could go through the pore one at a time. But on top of that, we know from bench experiments, and you'll see in this all-atom simulation, that the DNA threads through the nanopore with the bases in single file order. This is work by our colleague Alec Axementia at the University of Illinois in Urbana-Champaign. So right now, at first you see the DNA captured in a vestibule. The field is relatively weak. The main field is right here, and you can see that the DNA is elongated and base by base threaded through the nanopore. So how does ionic current vary? What about sequence identity accounts for that? And Deamer, in his original idea, thought that it might be based on simply the volume of each of the bases as they pass through the pore. And that proved not to be the case. This all-atom simulation shows what we think is going on. So the purple is our nucleobases, the yellow are the sugars, and the green is the protein. And you can see that occasionally the bases stack with one another. Occasionally the bases adhere to the protein surface, as does the ribose or deoxyribose. And at the end of the day, what appears to be going on is that if the DNA is sort of adhering to the side of the protein wall, the amount of bulk-like water in the nanopore orifice is relatively large. So that's depicted here. So the ionic current is relatively large. And depending on sequence identity, that can be blockaded. So the nucleobases basically occupy the aperture, and so the current goes down. So it's basically whether or not there is much bulk phase water in the nanopore orifice. So this slide basically condenses 20 years of work. Our lab was working on enzymes to regulate the movement of DNA through nanopores, initially with a DNA polymerase motor. And along with our colleague at the University of Washington, Jens Gunlock, who developed a more precise nanopore, protein nanopore, MSPA, as the sensor. It had better resolution. So the two of us got together, our two groups, and in 2010 to 12, demonstrated that you could read a sequence of synthetic DNA passing through the pore. The strand was basically a trimer of TAC repeated. And so as you see, there's a pattern, a trifold pattern throughout. G's will cause this to deflect. There's reasons for that I haven't got time to describe, but you can clearly see that this pattern was repeated over and over again. So that was the first evidence in the academic sector that one could read a DNA strand with a nanopore. Meanwhile, beginning in 2009, Oxford Nanopore had begun to work in anger, as they say in England, on strand sequencing. And this resulted in a seminal talk by Clive Brown, their CTO, at AGBT in February of 2012. So what in their technology, what is it based on? A lot of in very sophisticated implementations. A chip architecture that could support a thin film being shipped across the country or internationally by FedEx. Typically, those thin films are very unstable, and they develop an architecture to support them. Rather than using phospholipid bilayers, they develop synthetic films. Nanopore devices now can be stored for months to years and are still functional, and that would be very unlikely with a phospholipid bilayer. And lastly, they developed arrays of protein pores and motors that were bespoke, basically very, very clever protein engineering to optimize the motor that controlled flux and the pore that read molecules moving through the nanopore. This is a video from ONT, and I'll talk a little bit about it before I go on to the animation. This is the protein pore. It depicts alpha-hemolysin. In fact, the pore they use now is not alpha-hemolysin. The green is a helicase motor. And what you'll see is the helicase motor unwinding the DNA strand as it's fed through the nanopore. It also acts as a brake. It basically regulates movement to the millisecond timescale. And then on the right, it'll show the ionic current pattern, a depiction of the ionic current pattern that a neural network uses to decipher, a trained neural network to decipher base identity. So here's an animation. It shows one strand being captured. This is basically looks about like real time, thousands of nucleotides in no time at all. You can then reload with another strand and it reads automatically. So that's the basic idea. So let's talk about the present and future of nanopore sequencing. One thing I want to note is the next speaker, Sarah, our colleague Sarah, is going to talk about direct RNA sequencing, which is one of my great passions, in particular tRNA. And she'll talk about the various virtues of nanopore sequencing for this type of application. Getting back to genome sequencing, first let's talk accuracy. So when this first came out, and there was a community that were early adopters, and the sequencing accuracy was only 2 out of 3 bases. So it was a little bit painful initially, but over time they improved the HMM and made it more accurate to 70%. But the big breakthrough was principally due to using deep learning neural networks to improve the training sets. And that up to about 2019 had improved to about 9 out of 10 bases called properly relative to a reference. That improvement has not stopped. A combination of improved neural networks and protein design have brought this up to exceeding 98% in our hands for the median, as depicted here with this red figure, this red distribution, and over 99% modal. And one of the major developments in Oxford Nanopore executed was a reading head, 2 reading heads rather than 1. And the advantage to that shown on the right side is that you could read homopolymers up to 10 nucleotides in length based on the distance between those 2 reading heads. So the accuracy has gone up and it will continue to go up over time. Read lengths are one of the features. Mark talked about this, and a nod to PacBio, by the way, who's also helping lead the way in long read technology. This in nanopore space, it's not uncommon to get million base long reads. Interestingly, the base call accuracy for the first base is basically equivalent to the last base. So there's no fallout with greater length. And just to give you an idea of the scale, if you take the nanopore, which is about 85 angstroms long, the modern version, and you scale that up to the size of a hand, a 1 million base long fragment would be about 3.2 kilometers in length. So the DNA that is being sequenced dwarfs the nanopore. And importantly, now it's very easy with a single prometheon flow cell to get 10-fold coverage of a human genome in excess of 100 kilobases in length. And this greatly simplifies assembly of complex regions. And here's just a list. If you have a cell phone or a camera on your laptop, you can take an image of this, or I can forward these citations to you of a variety of ways in which this long-read technology has been implemented. I think one of the features of Nanopore, of the Oxford Nanopore devices, is its mobility. And there's no better illustration of this than putting a 90-gram mini on the space station. The image at left I think was taken in Germany. It shows, I believe, the stripe at the top is the space station, and the more faint stripe down below is the SpaceX capsule approaching that, and to basically hand off this mini. And it's a photo I like just for no particular reason other than that it's historically important. And this is astronaut Kate Rubins on the right, who sequenced DNA in space for the first time using a mini. And initially, they used DNA of known identity. I think it was lambda DNA. But now they're sequencing RNA and DNA of organisms that are on the space station. It turns out the space station, to my surprise at least, is not a surgical theater. It's not pristine. There's all sorts of things growing on the space station that need to be identified. And they can do that with MinION. And lastly, I want to talk about speed. And it's best to illustrate the speed with two examples. The first is our colleague Charles Chu at UC San Francisco. And he was able to use MinION from sample to result to confirm the first US case of Omicron infection, the 1st of December, 2021. And I think in infectious disease space, this is going to be phenomenally important over time. In part, the combination of speed and mobility. There's nanopore sequencers all over the globe, from the Arctic to Southern Africa. But getting back to speed, I think probably for me personally, the most impressive work of late was accelerated whole genome sequencing for clinical applications. And I don't have time to go over this in detail, but it was an effort led by Ewan Ashley at Stanford University, and there was support from Oxford Nanopore, our campus, Google Health, NVIDIA, and Baylor School of Medicine. And so what they did was they took the Prometheon, which has 128,000 nanopores. It weighs only 28 grams. So in fact, the first device they used, we put in our trunk and delivered to Stanford for their first initial analyses. And it reads at about 3 billion DNA bases in 3 minutes, which means you could get the sequence at 60x coverage of a human genome in about 3 hours. So they asked the question, can this expedite clinically relevant whole genome sequencing? And the answer is basically yes. On the bottom in the purple bar is sort of standard of care, what it took in the day to get a whole genome sequence for a diagnosis of a patient's condition. Took on the order of 56 days. Advances by Baylor and by the Ratty Institute in San Diego reduced this to a few days. These are what the Stanford folks referred to as the advertised times that Ratty, for example, could sell to a customer, to a hospital or what have you. And then Stanford had reduced this time to under 12 hours. And when I've talked to the people who basically engineered this, it's interesting, they would try to tweak the smallest of things, like somebody with a better software, a few lines of code, would pull out 5 minutes of less time. Somebody else would improve the library prep and get 37 minutes. But the fundamental reason why this went so quickly is that the sequencing is essentially real time. And using the cloud, they were able to make base calling and alignment real time as well. So that's really fundamentally why it got down to 12 hours. And there are more than 30 patients, I think, that they use this technology for.
2 minutes, Mark.
Yes, okay, I'll be done just a second. And one of the most dramatic cases was of a teenager from Portland who had presented with cardiogenic shock. And I am not an MD, so I'm not going to go through the details of this, but basically they need to determine whether it was a case that it was due to an inflammation of the heart muscle due to a virus, or if it was a genetic cause. And they used the Nanopore whole genome sequencing to identify a mutation in troponin, which is associated with heart muscle function. And if you're interested, a student, you may want to take a screen grab because these 3 papers basically describe this, this amazing technology in detail. So I want to especially thank NHGRI for their help. Like I said, I think earlier, they basically sponsored nanopore sequencing for over 20 years and stood by the technology when there were a lot of skeptics, and a host of different collaborators worldwide. Thank you.